Hey learners, welcome to Ahmad Coaching and I'm your teacher Dr. Anam. Today we are going to study about homeostatic function of liver. Hepat is the word which is the root word for the liver. So whenever you are going to hear about hepatic word, it means it is referring to liver. So if you are looking for the scientific word for the liver, it's going to be hepatic. Anything related to hepatic vein, hepatic artery, it means that it is related to liver. Liver is a vital organ in human body. It is located in the right upper quadrant of the abdomen, just below the diaphragm. Here you can see this is the structure of human body in which it is divided into 4 quadrants, like this. So liver is present in the right upper quadrant here, just below the diaphragm, which is this. It is the largest internal organ and the central station of metabolism. Maximum amount of chemical reactions or metabolism takes place in the liver. That is why it is also called as metabolic clearing house. That any byproduct which is formed during the process of metabolism, it is processed in the liver and removed from the body. It performs numerous essential functions critical to maintaining the homeostasis and overall health. Main homeostatic functions of liver include synthesis, storage, conversion, recycling, and detoxification. Let's study all these functions one by one. Starting with the synthesis, the first important function of liver is synthesis of plasma proteins. Plasma is a part of blood and plasma proteins, they are present in the plasma. Here you can see this is the blood. This blood indicates the plasma. The plasma, it contains plasma proteins. The first plasma protein includes albumin. It is the most abundant plasma protein and the function of albumin is to maintain the osmotic balance of the blood. Osmotic balance means that how much water should be present in the blood is controlled by the concentration of albumin. If the water is high, the albumin concentration is maintained somehow so that the water balance is maintained. The second plasma protein includes clotting factor. Clotting factors such as fibrinogens, prothrombin. These are the names of the clotting factors. They are essential for preventing excessive bleeding and ensuring proper wound healing. So whenever you have a cut anywhere on your body, these clotting factors, fibrinogen and prothrombin, they move to the injury site and they are going to make a clot over there. Clot means that they are going to seal that cut so that loss of blood is prevented. And liver is involved in the synthesis of these clotting factors. The third type of plasma protein includes transport proteins. Liver also synthesizes various transport proteins, aiding in the distribution and availability of essential minerals and vitamins. As the name indicates that they are going to transport something. Transport means they are going to take something from one place to another place. That is why they are called transport proteins, and these transport proteins, they are synthesized by the liver so that they can take minerals and vitamins from one place to other place where they are required. Second synthesis function of liver includes synthesis of bile. Liver produces bile acids and salts from the cholesterol. Bile is stored in the gallbladder and then released into the small intestine to aid in the digestion and absorption of dietary fat and fat-soluble vitamins. In the digestive system, you must have learned about bile, which is present in here, the gallbladder. The function of this bile is in the emulsification of fats. Itself, bile is not involved in the digestion, but what is going to do that it is released into the small intestine where it is going to act on the fat, going to emulsify it or convert it into small droplets so the enzymes they can act on it and digest the fat. So this bile is originally synthesized in the liver and the synthesis is done by the help of cholesterol. Means that cholesterol is converted into bile and then bile moves from the liver to the gallbladder. It is stored here, and whenever needed by the small intestine to digest the fats, it is released from the gallbladder, enters into the intestine, act on the fats so that it's emulsified or converted into small droplets, so that enzymes they can act on them and digest them. Along with the fats, they are also helpful in the fat soluble vitamins, which are A, D, E, and K. The third synthesis function of liver includes lipid metabolism. In the lipid metabolism, the first thing is cholesterol synthesis. The liver synthesizes cholesterol, which is essential for the formation of cell membranes, steroid hormones, as well as bile acids, as we just studied. So all these things, cell membranes, steroid and bile acids, they require cholesterol, and that cholesterol is synthesized in the liver. Then comes the lipoprotein production. Lipoprotein means lipid and protein combined together. The liver produces lipoproteins, which are very low density lipoproteins, low density lipoproteins, and high density lipoproteins that transport lipids, which are triglycerides and cholesterols. These are the names of the lipids, triglycerides and cholesterol, through the bloodstream. So in the form of lipoproteins, these triglycerides and cholesterols, they are transported from the bloodstream to the part of the cells where they are required. Proper lipid transport is crucial for energy distribution and storage. So this is how liver is involved in lipid metabolism. Then comes the fourth part of liver synthesis, which includes nitrogenous waste. Nitrogenous waste mean that compounds which contains nitrogen. The liver deaminates amino acids. Deaminates mean that they are going to remove the amino group. Here we can see this is the structure of amino acid. In the amino acid, with the carbon center, carboxyl group and amino group, they both are attached. In the process of deamination, this amino group is removed from the amino acid and is converted into ammonia. This ammonia is then entered into the urea cycle. Here you can see this, which is then excreted by the kidney. In the urea cycle, urea is formed, and this urea is excreted out of the body by the help of kidney. This detoxifies ammonia, a toxic byproduct of protein metabolism. So if you are taking excessive protein, which is more than the requirement of your body, what the body is going to do, that it's going to send that excessive amino acids to the liver. In the liver, the amino group from the amino acids is removed and is converted into ammonia. This ammonia then enters into the urea cycle, and through several steps, this ammonia, which is the most toxic byproduct, is converted into urea, and this urea, which is a less toxic product, is removed out of the body by the help of kidney. Second homeostatic function of liver is storage, that it's going to store something. First of all, it's going to store glycogen. Liver stores glucose in the form of glycogen. We know that glycogen is a long chain of glucose. This stored glycogen is crucial for regulating blood glucose levels. For example, during the period of low blood glucose, such as between the meals and when you are fasting and you are not eating anything, it means that the glucose level in your blood, it's going to decrease. At that time, liver is going to break down glycogen and it's going to convert this glycogen into glucose by the help of glycogenolysis process, in which glycogen is going to lysis or mean breakdown. So this is the process name glycogenolysis, in which glycogen is broken down to form glucose, and then it is released into the bloodstream, and this process happens in the liver. This process ensures a steady supply of energy, particularly for the brains and muscles, thus preventing hypoglycemia. Hypoglycemia means less glucose in the blood, hypo means less, And glycosemia means glucose level. It means that it's going to prevent less glucose condition in the body, and it's going to support the normal organ functions. So here you can see we have glycogen, and this glycogen is going to be broken down into the glycolysis by the help of glycogenolysis process, in which the long chains of glucose, which are in the form of glycogen, are broken down to release glucose so that you can prevent the hypoglycemia. The second function is vitamin storage. Liver stores vitamin A, D, E, K, and B12, which are the essential vitamins. Essential vitamins are the vitamins which are required by the body, so that you have to supply them because they cannot be prepared by the body itself. So these vitamins, they should be included in your diet. That is why they are called essential vitamins. These vitamins, they are vital for various bodily functions. By storing these vitamins, the liver ensures their availability for metabolic processes. So liver is going to act as a storage house for these vitamins, and whenever body requires these vitamins and you are not supplying them from the outside in the form of food, so what liver is going to do, it's going to take some vitamins from the storage and going to send those vitamins to the place of metabolism where they are required, so that overall health is maintained and to prevent any kind of deficiencies. The third storage function of liver includes iron storage. Liver stores iron in the form of ferritin. This stored iron is used for the production of hemoglobin in red blood cells, which is essential for the oxygen transport throughout the body. So iron is the main part of the hemoglobin. If there is no iron, it means that different hemoglobin chains, they cannot combine together to form hemoglobin. So iron is actually the central part of these four hemoglobin chains, and when the iron is present, then hemoglobin is combined together and is able to perform its function, which is the transport of oxygen from one part of the body to another part of the body. Adequate iron storage ensures that body can produce sufficient red blood cells, preventing anemia. Anemia is a condition in which body has less amount of red blood cells. To prevent the anemia and maintaining efficient oxygen delivery to the tissues. The fourth function of liver as a storage is triglyceride storage. The triglycerides, as the name indicates, tri means three, glycerides, that it contains three glycerides. The glycerides, they are made up of one glycerol and three fatty acids. Here you can see. So when three fatty acids, 1, 2 and 3, they combine together with one glycerol molecule, they are going to form one triglyceride. The triglyceride, which are a form of fat that serve as a long-term energy reserves, because they are the fatty acids which are the energy sources, they are stored with the glycerol in the form of triglycerides, and whenever you need energy, they are broken down to release energy. During period of energy deficit, when the energy is less, these triglycerides, they are going to broken down into fatty acids and glycerols to provide energy. This storage function help maintain energy balance and support metabolic demands during fasting and increased energy expenditure. Whenever you are fasting, it means you are not providing anything, any energy source to the body from the outside. At that time, the body is going to use its own reserves, which are triglycerides stored in the liver. Similarly, if you are doing extensive exercise, which is the increased energy expenditure state, at that time you also need energy, and this energy is again provided by the breakdown of triglycerides stored in the liver. The third homeostatic function of liver includes conversion. Conversion means that liver converts one thing into another different thing. The first conversion is carbohydrate metabolism. Carbohydrates are the compounds which contain carbon, hydrogen and oxygen. The first carbohydrate metabolism includes gluconeogenesis. Gluconeogenesis is a process which converts non-carbohydrate source. Non-carbohydrate means that other than carbohydrate source, which includes amino acids, lactate and glycerols, into glucose. In the last slide, we studied that triglycerides, they are broken down into 3 fatty acids and 1 glycerol. Fatty acids, they are used for the energy, but what will happen with the glycerol? That glycerol is converted into glucose. Similarly, amino acid, we heard it is deaminated, but what about the rest of the part of the amino acid? After the removal of amino group, the rest of the part is again converted into glucose. So this process is called as gluconeogenesis, in which non-carbohydrate sources, they are converted into glucose. This process is vital during fasting or extensive exercise when glucose levels, they are low, ensuring the continuous supply of glucose for energy, particularly for the brain and muscles. The second metabolism is glucogenesis. Glucogenesis means that excessive glucose is converted into glycogen. So for example, in gluconeogenesis, we have a lot of glucose. This glucose is exceeding the amount of body. So what happens that the amount of glucose which is required by the body is utilized, while the rest of the glucose is converted into glycogen by the process of glycogenesis. This stored glycogen can be broken down when needed to maintain blood glucose levels, so providing a quick energy source and preventing hypoglycemia. Hypoglycemia, like I told you before, that it is a condition in which body has less glucose. So here we can see that this glucose is converted into glucose 6-phosphate by the help of glycolysis, and then this glucose 6-phosphate is converted into glycogen by the help of glycogenesis, and this glycogen is then stored in the liver, and this conversion process takes place in the liver. The last function of liver is detoxification. Toxic means that something very dangerous. Detoxification means to minimize that toxic substance or to remove that toxic compound. Harmful chemicals, including food additives, pesticides, alcohols, and drugs, are detoxified and removed from the body, thus assisting in toxic disposal. So, any toxic substance that you ingest accidentally or deliberately, it's going to be into the liver, and in the liver, through different metabolic processes, those chemicals, those harmful chemicals, they are converted into less harmful chemicals, or they are detoxified, and after that they are removed out of the body. So that was all about homeostatic function of liver. If you have any question related to this topic, or if you wanted to learn about any other topic, please let me know in the comment section, and I will see you in the next video.